Thank you for coming, everybody. I'm Jingi Yeo in Steininger Lab at Seoul National University, and this project is about discovering fold novelty and biome-specific proteins from a billion structures. We actually clustered the metagenomic predicted protein structure database, and we extracted some insights from it. And this project is not published yet, and it will be soon to be in preprint. Yeah. So let's get started with this. So protein structure predictors have revolutionized biology. So the Dean Mine and David Baker got the Nobel Prize from it. And due to these protein structure predictors, what we can do is that everything that we relied on protein sequence, we now can do the same thing or more with protein structures. And these tools expanded the protein structure universe. So the predicted protein structure database started from 300,000 entries in 2021, and it became 200 million protein entries at July 2022. And we also conducted the research clustering the database previously, and the value of using structure for the analysis is that it can reveal remote homologs. So what we found, the examples from that database are like this. We found histones and interferon proteins and CD4 proteins structurally similar in both in human and bacteria. So this project is published in Nature 2023, so you can look over it and you can also learn from that. And furthermore, beyond this, 606 billion metagenomic predicted protein structures was now released. The database starts from magnified context. This is a metagenomic context. And then people predicted genes from it. So we got billion entries. And then people do the sequence clustering to reduce the redundancy. So this, using ESM fold, people predicted the structures of these 6 billion entries. So they published the 606 million structures. And this ESM fold is language-based, language model-based predictors. And the database has only have PFAM annotation and biome annotation. So it's very dark and unknown protein database. So adding this protein database, we now have 800 million predicted protein structures. So our project is cluster this full protein structure universe, which is 800 billion protein universe structures. So let's see what's inside it. So this is the clustering pipeline. We started the concatenated database, 800 billion, and then we did the sequence clustering using sequence identity 30% to reduce the fragments, which is abundant in the database. And then we extracted the high quality protein representative from each sequence cluster using the PLDDT, and PLDDT is the confidence score that the predictors return by itself. And then we structurally cluster the good quality sequence representatives with fold-seq using parameters like e-value 0.01 and coverage 90, which means that we cluster them by their structural homology relationship in their full-length alignment perspective here. And we got 72.6 billion structures, and this is the core structure that we analyzed the metagenomic database. So let's start with the first story, which is biome analysis. So we found proteins specific to extreme environments. The question starts with this: can you find any biome-specific proteins? Because organisms adapt to environments by modifying their protein sequence, like central dogma. And then I said, the database has biome annotation, then can we find any extreme environment-specific protein structures from it? So first, we determined the 1.8 billion clusters that have enough biome annotations, because ESM atlas the biome annotations are not on all of the ESM atlas, and AlphaFold database is totally lack of it. So we removed the clusters that have less than 10 biome annotations, so that we define core biome clusters, 1.8 billion clusters. And then we calculate the lowest common biome of the clusters. For example, if there are annotations like this in one example cluster, the biome annotation is in a lineage format, starting from root to its specific region. Then we computed the lowest common biome. Then that lowest common biome is the possible narrowest region that the structure exists. And as a result, 1.5 billion structures are universal, found in root, as you can see here, and environment and host, and environment of the rest. And they are all extreme environments in environmental lineage: thermospring and non-marine saline and alkaline, which is hypersaline environments, and glaciers. So we want to focus on these clusters, these structures. So we first wanted to see which organisms are generating those clusters. So we computed the taxonomy LCA. First, we predicted the taxonomy of the ESM atlas because they don't offer that. We aligned the ESM atlas against UniRef 90 for eukaryotes and virus, and GTDB for archaea and bacteria. As a result, we got 423 billion taxonomy annotations. And then we computed the LCA, the organism that the cluster is conserved at, which means that the principle is like, if there's a member A with the taxonomy lineage like this, 1 to 3, and B, for example, 4 to 3, and then we can convert the taxonomy tree of them. And this shaded area is the taxonomy lowest common ancestor, so that the structure is conserved at 2. Then let's see extreme environment-specific proteins. First, thermospring. The thermoproteota is the most producer of those structures, and these are the examples. And these are only found in thermospring by our approach. DNA polymerase and thermosin found in thermospring. And next one is hypersaline, and halobacteria are the most producers. And fatty integrase and protein kinase, specific to hypersaline environments, and glacier, mostly produced by cyanobacteria, and horizontal transfer protein and caspase have specific structure in that environment. Yep, and let's go to the next story. This is about domain analysis. How the ESM atlas, the metagenomic database, expanded the protein structure universe. Firstly, we removed the singletons, the cluster that have only one entry. As a result, we got 5 billion clusters, and then we removed the cluster that have at least one AlphaFold database entry in it. So these clusters are unique structures in ESM atlas. Then this might have expanded the protein structure universe. Then what are they? So the first approach and our first assumption was that they must have novel domains. So we determined novel domains starting from that 3 billion ESM-only clusters. We chopped the domains and determined 5 billion domains, and we removed the low-quality domains using PLDDT, globularity, and secondary structure. And then we searched them against all of the domain databases so that we determined 15,000 domains. And then we measured the novelty and credibility by our tools. We determined 2 novel domains. And these are the novel domains. One is non-symmetric and one is symmetric, and they are all from bacteria. But this number is very few, and then what are the remaining majority? So our next approach is that then they must be a combination of existing domains, but their combination is novel. So we studied again from the ESM-only clusters. We found that approximately 400,000 multi-domain chains, multi-domain proteins are there. And then we focused on removing the redundancy. We determined 49,000 combination of domains and researched them. If we only found the combination, never found in AlphaFold database. So the novel domains is 4.4K novel domain combination. And these are the examples. These are the top 10 frequent pairs in the novel multi-domains. You can see the TONB receptor. This is the transmembrane receptor, and periplasmic function are repeatedly found here. And this is one example of the novel domain combination. TONB receptor is here, and its plug domain is here, and one enzyme is here. And please keep in mind that these combinations have never found in AlphaFold database. Yep, so the ESM atlas has expanded the protein structure universe. So as a conclusion, Sequence is at the core of most protein bioinformatics. So we did a lot of things with that. And then now we can supercharge analysis using structures. So as a summary, we structured the entire protein structure that are over 800 million proteins, and we identified 1.8 biome-specific clusters, and extremophils are mainly producing extreme environment-specific proteins, and we identified 2 novel domains and determined 4.4 thousand novel multi-domain combinations. And in the same group of the previous speaker, and we collaborated with UCL, and thank you for, I appreciate all of the collaboration and contribution. And we have the recon next spring, and I'm making homepage here. Please visit Korea and the recon, the computational biology conference. And this is my talk. Thank you for listening. Thank you.
Thank you. Is there any question or comment? Okay. You find extreme condition specific fold in alpha fold structures. May I see that fold again?
Slide. Yeah.
Do you have any idea why this fold structure
Yes.
is fit to the extreme conditions like high temperature or high ionic strength?
Currently, I don't know why, but they have when I try to align them structurally to other structures that have similar function, and they have unique, say, some deviation in their structure, like having one more alpha helix or one more IDR at the unexpected region. I think that affected its, say, rigidity to stand this condition, I think. So far, yeah.
You mean that these folds are larger, tend to larger or smaller than the normal ones?
In a length?
Size, yeah, size.
I didn't look over that yet.
Okay.